Hello, my name is Ben Dribus. I'm a math professor at William Carey University in Hattiesburg, Mississippi, and a member of the Foundational Questions Institute. This is video number 1 in a series of short videos demonstrating advances in artificial intelligence I've been working on, involving novel artificial neural networks for use in science and medicine. The goal of these videos is to help establish collaborations and partnerships with others who are interested in developing or using this technology. You can find my contact information on each slide. I will begin with a brief preview of this technology using the Python programming language in Jupyter Notebook. This is a medical data analysis problem involving breast cancer diagnosis. These images describe the physical details, which I'll discuss later. I'll load some raw data about the problem, then pre-process the data. This cell here does both those tasks. Now I'll construct a neural network to analyze the data. Here's a visualization of the network, neurons and synapses, which I'll come back to. Next, I train the network. This takes a few seconds so I can explain what's happening. The scrolling numbers are the percent error, that is the number of patients out of 100 that the network misdiagnoses. We want that number to go down close to 0. It's often good to train in bursts called epochs. Between epochs, the network is modifying itself. Now, this isn't just the usual changing of synaptic strengths. It's actually completely removing and adding connections. You see there it actually got down to 0% error. That's partly luck, honestly. It wouldn't diagnose every set of patients perfectly. In fact, right now it looks like it may be losing accuracy, but I've written the software to save the best configuration. At the end, I have it print a graph showing how the error went down, so we see the network diagnoses well after training. Finally, I'll show you how the network modifies its architecture during training to become more highly connected. This looks messy, but then again, so does your brain. There's a lesson in this which I'll explain as we go along. Now, before further exploring what we just saw, here are the topics for the video series. All of these can be done on a single ordinary CPU core. This first video demonstrates construction and training of a novel neural network and introduces my collaboration interests. Video 2 will explain novel network architectures graphically. Video 3 will examine the breast cancer problem in more detail. Video 4 will examine a classic image classification problem involving handwritten digits. Video 5 will demonstrate general network-based disease diagnosis. Video 6 will tackle a physical chemistry problem. Video 7 will involve MRI data. Video 8 will explain my collaboration interests more thoroughly. And video 9 will discuss the references for these videos. Let's return now to the breast cancer problem. This data set has 569 patients with 30 cellular measurement indicators for each patient, taken from cells like the one shown in the image. The goal is to diagnose malignant or benign from these indicators. This data set comes from the University of Wisconsin. It can be found on the well-known UCI machine learning repository. It's well established. Now, medical professionals tend to ask me detailed medical questions about how this data is gathered. The method is called fine needle aspiration. Cells are drawn with a needle and imaged microscopically. The 30 indicators are measurements taken from these images. This is simpler than analyzing the images directly. It's a standard, minimally invasive multi-tissue biopsy method. These images are free from Wikimedia Commons. Let's look at the basics of training a diagnostic network with this data set. Here's how the network starts out. The colored nodes represent neurons and the edges connecting them represent synapses. The 30 indicators are fed into the 30 green input nodes at the bottom, and the diagnosis, malignant or benign, comes out of the 2 red output nodes at the top. We show the network the correct diagnosis for 469 patients, called the training set, then test to see if it can accurately diagnose the remaining 100 patients, called the testing set. My training method is a modification of the popular ADAM method, which I call adamnesia for its selective memory properties. The network modifies itself by pruning and adding edges, not just changing the weights, or synaptic strengths of the edges. In this case, I choose to keep the total number of edges constant. The network starts with a split hemispheres effect, but it learns to repair this deficiency so that it's 100% connected. Here's how the network looks after self-modification. Now this graphic shows that self-modification does improve performance. Red is the average performance of the self-modifying network, and blue is the weaker performance of a static network under the same conditions. These performances are averaged over 100 trials. Any individual network tends to do better than this somewhere in the training process, and some even reach 0% error as we saw. So this is not really a hard problem, but it's useful for illustration. The machine learning site Kaggle has other examples with this same data set. Now that we understand the problem better, I'll show you a fleshed out version of the network construction and training process. This is an expanded Jupyter notebook. It may occasionally take a second or two to scroll. These cells involve the raw data. You can look at individual rows, for instance. Now this schematic illustrates the different roles of training and testing sets. It's important to emphasize that the trained network is not merely regurgitating answers it's been trained with, because it's tested with completely unfamiliar data. I'll skip the details of data pre-processing. Next comes network construction. The details of this are also for a later video, so I'll move on to network visualization. Here's the original network architecture I showed you before. You can see the split hemispheres completely dividing the network in half. Now, why did I design it this way? You might argue, well, of course it diagnoses better after self-modification because you started with a silly architecture. The answer is that I chose the split hemispheres to illustrate in a visually obvious way the poor connectivity properties of a network that is too local, with not enough edges reaching far across the network. In the picture, local edges are the ones connecting nodes to the closest nodes in the next layer. All the edges in this picture are local. Non-local edges appear here in the self-modified network. They reach far across the network. Now, it turns out that this same basic structural problem of too much local structure and not enough edges reaching across the network occurs in the most powerful type of networks in use today, called convolutional neural networks. Now, AI is a multi-billion-dollar industry, and the really smart people who design these networks know about all this. So why do they design them that way? Well, oftentimes interesting data, such as images, has local features that require local network structure to analyze them. This calls for both good connectivity properties and local structure, and these pull in different directions. The standard solution to this problem is to make the final part of the network very dense and highly connected, like here in this illustration. You see down here the network is completely local with the hemispheres split, but up here every possible edge is included. But this kind of density is computationally costly, and such networks can take months to train. What we really want is networks that are A, sparse, that is, not dense, B, local, and C, highly connected. Well, this is precisely what my novel networks are like. Furthermore, self-modifying networks seem to rearrange themselves in this way if given a chance. And yes, I know this is an ugly duckling of a network, but if you think about the brain, or the web, or the Hubble deep field, or space-time at the fundamental scale in all probability, it's in pretty good company. We'll see all this carefully illustrated as we go along. These cells show network parameters. In particular, the connectivity starts out at exactly 1/2 due to the split hemispheres. Now we come to network training. This schematic shows how the training process works, but I'll skip the details for now. This cell contains the training parameters. For example, these two parameters determine how edges are pruned and added. This time I'll start and then quickly interrupt the training process to illustrate something. I'll let it run for just one epoch here. This will take a second to finish. Now I'm interrupting it. You see, this is what you get when you do a keyboard interrupt. You can see here in the illustration that the network has just started to stitch together the split hemispheres, and the connectivity has increased to 88%. Now down here is an illustration showing what happens after many epochs. The split effect is gone and the network is totally connected, but most of the structure is still local. So we see a natural migration toward a sparse, local connected architecture. My overall message is that existing technology can be significantly improved by using these novel network architectures. To reiterate, they are A, sparse and therefore efficient to train, B, local and therefore able to analyze local data features, and C, highly connected without using costly dense layers. These are pages from documents where I developed this theory. Now these improvements combine ideas from biology and physics. Biological inspirations are routine, like how the visual cortex inspires convolutional neural networks. Self-modification is biologically familiar, for example. But some of the physics ideas may be less familiar. For example, unless you're interested in astronomy, you may not have studied the horizon problem and inflationary cosmology. But in a network setting, these ideas shed light on the perceptive field. That is, how much of each layer you can see from a given node as you move back in time through the network. Ultimately, this involves sophisticated geometric ideas. Now, an obvious question from the outset is why not just use existing machine learning packages like Keras and TensorFlow? Well, for routine things, these packages are great, but they're not flexible enough for the highly customized layer structure and detailed training control that I need. I'll wrap up this first video with a preview of my collaboration interests. Here's my contact information again. So what am I looking for? Well, first, interesting data sets to analyze. If you represent a company or research group needing data analysis, then I'm interested in talking to you. Second, software development. I want to speed up and generalize my code. These ideas work, so this would be a high reward project. Third, computing time. I've proven mathematically that my methods will scale well for very large data problems, but this will ultimately require supercomputing. And fourth, working partnerships to make these things happen. Now, partnerships with whom in particular? Well, first, medical groups such as hospitals, medical schools, even individual doctors with interesting data. I'm quite interested in contributing to practical medicine. Second, any technology groups using AI. I can offer specialized network design capabilities that you won't find elsewhere. Third, any researcher working on interesting data problems. Research papers on this will practically write themselves. And finally, students can participate in a lot of this, and it could be an excellent resume item. Thank you so much for watching. In the next video, I'll carefully explain the meaning of novel neural network architecture with lots of graphics. If you're mostly just interested in applications, then several subsequent videos will provide demos involving specific data analysis problems.